Hey, welcome to part two of the lecture on chapter 16, adaptive immunity. We just finished off with lymphoid organs, and we're going to move on into the term antigen. So antigen means antibody generator. That is, these are structures that elicit the body to make antigens, it elicits the body to make antibodies. In other words, all those like little arrow structures, anything that's going to bind to one of those little arrow structures is what we would refer to as an antigen. So they're molecules that the body makes. Foreign antigens are things that the body could recognize as foreign and worthy of attack. Um, and then antigens like the bigger structure, and then they're like specific little knobs or buttons or like pokey things that the antibody actually touches on or connects to. You'll hear this word epitope. I'm not going to make a big deal about that word, but it's a piece of the antigen that the antibody can react to. I'll give you an example. If it's a foreign macromolecule, something that's different from us, actually, this is a really important point. Our immune system can recognize things better that don't belong here when they're quite different from our own bodies. You know how we were talking about how do you get like the best antimicrobial drugs? It's based on things that are different from our structures. Yeah, so that way you can attack something without hurting your own tissues. And that is what our immune system has been doing for hundreds of thousands of years. So some of the things that our immune system can react to are pieces of bacterial cells, like, for example, cell walls, but also things that are unique to viruses or fungi or protozoa, things that are different from us. Now, here's the tricky thing. Sometimes our immune system is a little bit problematic, let's say, and it'll overreact to stuff that's not actually dangerous to us. Those are the things that we refer to as allergies. So sometimes foods that are not dangerous, like peanuts, strawberries, shellfish. For some people, those things are deadly because their immune systems react to it in a way that you would think the world was ending. And other people, it's just an inconvenience. They might have like seasonal allergies. Same deal. It's the immune system overreacting to something antigenic, something the antibodies can bind to. All right, so what I'm talking about, antigens can be any structure that's large enough that something can bind to. And then this is a zooming in on the structure. Do you see those tiny little bits that are a little funky shaped that our immune system can react to really strongly? That's what sometimes you'll hear referred to as an epitope. You'll also hear the term antigenic determinants. It's almost like they want us to not understand this stuff. So if you hear the word epitope or antigenic determinant, you can substitute antigen, like it's a thing that I'm going to react to. All right, types of antigens, things that are outside of us, things from microbial cell walls, other structures, flagella, etc. And then endogenous antigens would be things that our immune system can react to that were actually inside our cells. They were produced by microbes, like, for example, viruses, but they're inside our cells. And then autoantigens are things that our immune cells can detect, but they're from us. They're from normal cellular processes. All right, so what I'm talking about, extracellular microbes, we've got exogenous antigens, so the thing is still outside of the cell, like a bacterium or some virus particles before they've gotten inside the cell. But let's say the virus particle got inside the cell. The cell is now infected, it can present little pieces of this virus, present that on the surface. It's a kind of way of asking for help, if you will. But I'll have to explain something else about the immune system first. And then here's a normal cell kind of showing kind of normal stuff on its outside. All right, I'll tell you about it now. If you can you picture being pulled over by a police officer? And then the very first thing, I want to see your driver's license and registration. Like, you haven't necessarily done anything wrong, but we want to see your ID and some other documentation that you're doing things correctly. If you have trouble producing those, or if you produce something incorrectly that doesn't look right, now we're getting into a problem. That is what happens when the immune system is cruising around, just looking. The cytotoxic T cells are just cruising around, just checking everybody out. And they're basically always asking for driver's license and registration, please. And driver's license and registration, everything is intact over here, but this is looking funny. These are not things that we have in our body. Usually we know that there's trouble here. I mean, I think of this as, driver's license and registration, and you pull out a note that says, help me or kill me, please, because I'm a danger to the rest of the body. And so then this cell can get taken out by the cytotoxic T cell. All right, more about that later. All right, so preparation. Let's back up. You see how these things are being presented on the outside of the cell? Those structures on which the cell is presenting material has this really cumbersome name. And it is called a major histocompatibility complex protein. Man, I know that's terrible. Believe it or not, you've actually heard about these before, whether you recognize that name or not. You learned about these if you've ever heard anything about an organ transplant and trying to find a match, a donor with a match and a multiple point match. This is what they're trying to match, are these major histocompatibility complex proteins, these things that are going to be presenting things on the outside of the cell. And the more similar we can make, or we can find a donor to the person who needs the transplant, the better off they're going to be, because their immune system is not going to be trying to kill this organ all the time. And we'll talk about these presentation platforms, the major histocompatibility complex, and what they have to do with the antigen presenting cells. Well, I've already mentioned determining compatibility tissues for tissue grafting. Originally, they were described in humans. Then they were discovered in all kinds of vertebrate animals. Essentially, they are the platform on which the cell will hold and position material for the presentation to the immune cells. So again, think driver's license and registration. I'm going to present it like this, because the cop that's coming to see won't be able to see the driver's license and registration if it's just put out willy-nilly. It has to be presented in a certain way, and the certain way is on these major histocompatibility proteins. So here's the antigen presenting cells presenting the stuff on an MHC protein to, in this case, a B cell or a T cell. And yeah, the Sistine Chapel, it's a little biology humor for you. So yes, they make contact. The B cells or the T cells, they are cops, but like, they can't observe things for themselves. They have to be shown stuff. And they have to be shown stuff on the major histocompatibility proteins. The role is the antigens. Okay. The antigens get sat upon these platforms to present it to the cops, to the T cells and the B cells. I'm not going to go into huge amounts of detail on this. We're going to talk in general about the MHC1 and the MHC2, and different COPs need different platforms to look at. All the cells of the body present on MHC class 1. And it's the cytotoxic T cells that can look at that one. And MHC class 2, those are unique to the antigen presenting cells. So when a macrophage eats something and then it says, oh, I got to go tell somebody about this, and it leaves the site of infection and it goes into the lymphatic system and it goes to present this, it's presenting it specifically to the helper T cells. And the helper T cells can read things or can notice things that are presented on the MHC class 2. That's the big difference, is all the cells in the body are presenting on MHC class 1, and the phagocytic cells that then go and present this to the COPs on purpose, this is on class 2. In general, it's just a platform. It's embedded here in the cytoplasmic membrane. Remember the phospholipid bilayer? It's embedded here. The important bit is that there's a groove where the antigen's got to fit, so it's got to be presented properly. And so examples of antigen presenting cells are these dendritic cells, for example. And there it is presenting. I'm going to play this little video and then we'll be done with this section of the lecture.
Special antigen-presenting cells, including monocytes, macrophages, and their relatives, engulf invading pathogens and digest them, producing fragments with antigenic determinants.
Okay, remember that antigenic determinant is just a fancy way of saying, I'm going to show some antigens now. I'm going to show you this is the stuff I, as a macrophage, am going to present this to a helper T cell to get the helper T cells to help me out with this trouble. Again, with the cop analogy, it's the local police then getting some fingerprints or something and then showing it to the FBI and saying, this, if you see this, this is what we're going after. And we need to process it. We're going to take the fingerprints, or we're going to, I don't know, take pictures, we're going to do something, and then we're going to present it in a certain way. And that's what's happening in here. And you're going to see the MHC proteins there. I think they're shown as like these green loops. Those are going to be the platforms on which this gets displayed on the outside of the cell.
Vesicles containing MHC proteins fuse with vesicles containing digested pathogens. The antigenic determinants from the pathogens are loaded onto the MHC proteins. MHC proteins are used to present peptides on the surface of the cell. Since the peptides are produced from proteins inside the cell, they allow the cells of the immune system to detect proteins inside the cell without entering the cell.
Okay, so think I've got fingerprints and mug shots, and I'm going to present them. And you don't have to come inside to see them. I can just show you out here.
Cells in the body routinely break down proteins into small peptides, which are about 8 to 10 amino acids in length. These peptides enter the endoplasmic reticulum and are loaded onto MHC proteins. The vesicles containing the MHC peptide complexes bud off of the endoplasmic reticulum and are released into the cytosol. Vesicles containing the MHC peptide complexes travel through the cytosol to the cytoplasmic membrane, delivering the complexes to the exterior of the cell.
Okay, so this is like, I'm just going to have images of my driver's license and registration just out here all the time, just in case some police come by. And the police come by all the time, and they're always asking for driver's license and registration just to make sure that you're supposed to be here. And that's the cells, I'm pointing with my finger, and that's the cells kind of showing driver's license and registration all the time.
If this cell is functioning normally, the immune system cells will simply ignore this presentation. However, if the cell is infected and thus presenting viral proteins, the immune system cells can notice the problem and take action.
So if when the cell is presenting, it's presenting not its normal driver's license and registration, but the registration's kind of wrinkled, or it's the wrong paper, or there's a note that says, help me, please, then the immune system can start dealing with the cell. And the same thing can happen if the cell is behaving weirdly, not because it's infected from the outside, but because there are genetic mutations that have occurred that are leading to the development of a cancer. Same thing. What's being presented on the outside is not quite right. And the immune cells can take care of this precancerous condition. And that is the end of part 2. See you over in part 3.